Yeah, but then I think it's time to start, and I want to welcome everyone to this last of our lecture series for this winter term. And yeah, I also told our guest today that I think it's a very sad coincidence on this yeah, sad day to talk about trauma on the one hand, while we have to experience that on European grounds we are now facing acts of war and aggression against the Ukrainian people. But on the other hand, maybe it's also and today in the morning I received maybe like some of you, I received this announcement by the ESTSS that said, well, we also as trauma researchers, we have to stand together and see what we can do to overcome trauma. And that's why I think maybe it's also promising to welcome today our today's guest, Professor Soraya Seedat, who is distinguished professor of psychiatry and holds the South African Research Chair in Post-Traumatic Stress Disorder. And yeah, Soraya, I'm very delighted. I feel very delighted that you accepted my invitation, because I very happy that I had the opportunity already in the past to collaborate with you in different ways. And you said you that I should not talk, make a brief introductions, but still I have to say that I'm very impressed by all the work that you are doing, and it's always a big pleasure when I have the opportunity to work together with you. And that's why I think it will be a very inspiring talk, and we are all very much looking forward to your presentation. And yeah, thanks again.
Thank you very much. So it is indeed a pleasure to be speaking to you today. I am very encouraged to be speaking about work that I have been involved with for some time. I just want to first check with you that you can see my screen.
I think it works perfect. We can see you and we can see the screen, and we hear you properly.
Okay, so thank you so much. So as Roland has indicated, my talk today is going to focus on understanding epigenetic and genetic mechanisms of trauma and PTSD in high adversity, low resource contexts. And that is because I live in a high adversity, low resource context. And Roland has afforded me a very kind introduction, and it is a delight to address you today. It is a sad day. I won't be focusing my talk on war-related trauma, but we'll highlight some of the long-lasting and negative impacts of exposure to trauma across the lifespan. So this is work that is fast evolving in the global field of trauma and post-traumatic stress disorder. Genetic and epigenetic signatures in childhood trauma and PTSD have become a very intensive focus of investigation. And what I'm going to do today is to share with you some of our work in a high adversity setting where the burden of trauma is particularly high, and it's juxtaposed against other disease burdens. So in South Africa, we have a quadruple burden of disease, and as I have just said, violence and injuries are juxtaposed against other three major disease burdens, which have received the lion's share of research funding in South Africa. And it has become particularly challenging over time, and even more so during the COVID pandemic, to secure funding to do large-scale genetic and epigenetic studies in the trauma field. That said, I've been very privileged over the past 15 years to be afforded funding from South Africa to set up a Genomics of Brain Disorders research unit. And for the past 15 years, I've also been chair of the South African Research Chair in PTSD. Now, our focus over the past 10 years has been to interrogate PTSD as well as other neuropsychiatric disorders, and to do this on a genetic and epigenetic level, and to use omic technologies insofar as we can with the resources that we have to better understand the biomachinery underlying PTSD, as well as other comorbid psychiatric conditions. So in my talk today, I'm going to highlight cumulative trauma and focus specifically on the impact of childhood trauma. I will then use 3 studies of childhood trauma with or without post-traumatic stress disorder to show how findings from these studies can enhance our understanding of the pathogenetics of cumulative trauma. And this is against the backdrop of research that has been done, particularly large-scale studies that have been conducted by the Psychiatric Genomics Consortium for PTSD. And I have been privileged to be part of this consortium, the work group for PTSD, for the past 5 years. And the 3 studies that I'm going to highlight exemplify the burden of disease that we have in South Africa and high-risk populations for trauma. And I'm going to specifically focus on adult rape against the background of early childhood adversity, HIV, also in the context of early childhood adversity, and then adults with post-traumatic stress disorder who also have medical morbidity. And in South Africa, we have, in the context of non-communicable diseases, very high rates of hypertension, diabetes, and other cardiometabolic diseases. So Judith Herman, who is a very well-known trauma researcher, has said that the conflict between the will to deny horrible events and the will to proclaim them aloud is the central dialectic of psychological trauma. And this rings true because there is a tension between wanting to forget and being overwhelmed by traumatic memories in those who have been exposed to trauma. The pervasive and harmful impact of traumatic events is heterogeneous, and this makes the field particularly challenging to try and determine what are the neurobiological underpinnings of PTSD, but also its comorbidities, as well as to more specifically try and unravel the genetic and epigenetic signatures of PTSD when these comorbidities are highly co-occurring. And this has been one of the challenges of the Psychiatric Genomics Consortium PTSD work group, trying to disentangle comorbidities such as major depressive disorder, anxiety disorders, but also medical comorbidities such as metabolic syndrome, in determining unique genetic and epigenetic motifs. Now, in addition to the effects of trauma on mental health, trauma can also have a direct impact on a number of physical disorders or physical conditions, independent of psychiatric disorders. And when we consider post-traumatic stress disorder and other psychiatric morbidities that emerge in the face of trauma, I think it's also important not to forget that many individuals experience subthreshold phenomena that may not meet DSM-5 or ICD-11 criteria for a psychiatric disorder, but may nevertheless experience substantial impairments in everyday functioning, as well as high levels of subjective distress. So PTSD is the quintessential trauma-related disorder, and it's a diagnosis that I think many of you will know has remained controversial since its inception. And compared with other mental disorders, such as major depressive disorder, bipolar disorder, schizophrenia, it is a relative newcomer in the psychiatric nomenclature. There is one feature that has remained constant over time, and that is the requirement that there be exposure to a trauma, and that is a qualifying trauma exposure. And that of itself has been controversial because there are many individuals who feel that the defining traumas for post-traumatic stress disorder are too narrow and should be expanded. And I think that has particularly come to light during the COVID pandemic, where there have been many studies that have come in the light of the pandemic, defining trauma as the pandemic itself. But suffice to say that, the feature that is at the heart of PTSD is its complexity and its heterogeneity. And we need to try and conceptually differentiate trauma and exposure to trauma, and then the psychological responses to the trauma, as well as other negative sequelae. So trauma and PTSD are highly prevalent in sub-Saharan Africa and not just in South Africa. And we know from studies as well as systematic review and meta-analysis that was published in 2020 that people living in sub-Saharan Africa are disproportionately affected by interpersonal traumas, but also other war-related traumas. So if we take the country that I'm in, rape is one of the highest, rape has one of the highest prevalences, both lifetime as well as 12-month prevalences of all of the interpersonal traumas, together with intimate partner violence. So South Africa is one of those countries that ranks very high in relation to rape and intimate partner violence prevalence. But we also have very high rates of other forms of physical and sexual violence. So if you look at the pooled prevalence in sub-Saharan Africa of post-traumatic stress disorder, so individuals who have been exposed to trauma and develop PTSD, from adult and youth samples that have been pooled in the meta-analysis that I mentioned, a rate of 22% has been documented. But we know that there is a mismatch, in fact, what we refer to as a treatment gap between the rates of PTSD and the receipt of treatment. And that treatment gap is very high in sub-Saharan Africa. So in South Africa, we have a 90% treatment gap for common mental disorders, translating to only 10% of individuals with PTSD or depression or substance use disorders, in fact, receiving treatment. So as I mentioned, interpersonal violence is associated with a very high risk of PTSD. That is not only true for sub-Saharan Africa, it is true internationally. And this has been replicated in studies over time. And what you see on the left of your screen is just a documentation of 12-month prevalence rates of PTSD across a number of high, middle, and low income countries. We know that most individuals exposed to trauma do not develop PTSD, but it is those who develop PTSD that we're most concerned about because they often have long-lasting symptoms. That has been shown in many studies, population-based studies that have been conducted internationally. It is particularly important that we try and identify the etiological underpinnings of post-traumatic stress disorder, because I think by better understanding who is at highest risk for PTSD, we can tailor our treatments accordingly and also optimize treatments, because we know that both the pharmacological and psychotherapeutic interventions that we have for post-traumatic stress disorder are not optimal for the vast majority of patients, particularly those who have chronic PTSD and who have other comorbidities. So against the backdrop of interpersonal violence, we also have very high rates of early childhood trauma exposure in adults who develop PTSD, but also in the population at large. So irrespective of a diagnosis of PTSD, childhood trauma is highly prevalent, and 60% of children are exposed to trauma by the age of 16 years. So much of the trauma burden that we see occurs in childhood and adolescence. And many children and adolescents who grow up as adults will have been exposed to multiple traumatic events. And this is what I am in fact referring to by cumulative trauma, although cumulative trauma has been differentially defined. And I think it's important to bear that in mind, that the heterogeneity in terms of defining cumulative trauma and complex trauma and chronic trauma has plagued our field. So as I said, childhood trauma is a global burden. In South Africa, about 42% of children have experienced some form of maltreatment. Much of it is in the form of sexual and physical abuse. Emotional abuse and emotional neglect have been less well studied. In addition to childhood trauma exposure, many of these children and adolescents experience daily victimization in their community. So criminal victimization, and in the province in which I reside, which is the Western Cape province, where Cape Town is located, we have very high rates of gang violence. So individuals living in low to middle income communities have daily exposure to criminal victimization, gang violence, and other forms of community violence. Now, as I indicated, only a small proportion of trauma-exposed individuals will develop PTSD, and that is true across the lifespan. So what is it that predisposes individuals to PTSD? And so we know that there is a conglomerate of factors that can explain the disproportionate burden of PTSD in high-risk groups. And women, for example, constitute a high-risk group. And there have been many reasons that are put forward, some of them that have been empirically validated, showing why women may be at much greater risk of exposure to post-traumatic stress disorder. And because I'm focusing on genetic and epigenetic factors, which really, as part of this conglomerate of etiology, is just one type of contribution. I think it's important to bear in mind that we now know that genetic factors may also explain the high heritability risk in women, and there have more recently been molecular genetic studies that have confirmed specific genetic variants, particularly a gene that is an important functional gene in the HPA axis, known as the polypeptide 1 pituitary receptor type gene, that may explain the high risk of PTSD in women. Now, when we think about etiology, I think we also need to think about pathways that link trauma exposure to morbidity and mortality. And as I said at the outset, I'm going to focus my talk and present data in high-risk populations, and more specifically in rape-exposed individuals in my setting, but also in individuals who have cumulative trauma in the context of HIV, as well as cumulative trauma in the context of post-traumatic stress disorder and cardiovascular disease risk. And what you see here is really just a kind of putative model showing some of the various relationships between early trauma exposure as well as adult trauma exposure, and some of the very pernicious outcomes that we see in the context of PTSD. At the top, a number of unhealthy behaviors that may also explain some of the pathways between trauma exposure and disease outcomes. And at the bottom of the screen, some of the mechanistic influences that have already been shown in many studies of different trauma populations with and without post-traumatic stress disorder. Now, in the context of omic investigations in post-traumatic stress disorder, but generally in the field of psychiatry, I think it's fair to say that the world's populations are not equally represented in GWAS studies. And in fact, there's a gross disproportion between the populations that have been investigated in genome-wide association studies and epigenome-wide association studies relative to the global population demographics. So this has been of grave concern, especially on the African continent, where African populations have been markedly underrepresented, and that is starting to change with large consortia like the Psychiatric Genomics Consortium, where researchers on the African continent are contributing data in the form of DNA, RNA, other biological samples to the consortium, as well as contributing clinical and other biological data to drive this endeavor to make these large-scale genome-wide, epigenome-wide, and other omic studies more inclusive of African populations. And we too, on a small scale, have also contributed more recently to this endeavor by conducting a genome-wide association study and then using polygenic risk scores to try and predict post-traumatic stress disorder and metabolic syndrome in a South African mixed race population. And mixed race as well as African populations, because they are genetically admixed, pose major challenges for studies of pooled samples internationally. But it is important that we have more African representation. So it's very encouraging that this picture is starting to change. So in fact, since 2016, there has been a flurry of activity to ensure that African as well as other non-Caucasian populations are better represented in these studies. So the work that I'm going to present today is primarily aimed at identifying genetic and epigenetic signatures associated with childhood trauma and or PTSD. And the reason that this has formed the thrust of our focus and investigation is that if we can identify genetic and epigenetic signatures in the context of the many environmental and other sociocultural influences that contribute to the development of PTSD and other comorbidities, we can better identify predictors of risk in adulthood, and this could in time drive more tailored treatment approaches. So we're not yet ready. I mean, this is not prime time. Internationally, I think the field of genetics and epigenetics and other omics in PTSD has lagged behind other psychiatric disorders such as schizophrenia and bipolar disorder. But I think that we are on the path to fast catching up. So if we think about trauma across the lifespan, we think about sort of complex trauma and cumulative trauma. Initially, many of the studies were candidate gene studies that identified a number of genetic variants across genes that are primarily involved in the HPA axis and in neurotransmitter systems and in the neurobiology of post-traumatic stress disorder, and they highlighted on this slide. So in addition to these candidate gene studies, as I mentioned, there have more recently been large-scale genome-wide association studies and epigenome-wide association studies. We think about genetics and we juxtapose that with epigenetics. When you investigate epigenetic changes, you are essentially looking at functional changes that may alter gene expression without changing the structure of the DNA. So epigenetic changes occur via two main mechanisms, and I'm not going to delve into these mechanisms, but suffice to say that the two main epigenetic mechanisms that have been investigated in PTSD are DNA methylation and histone acetylation. Now, until recently, there has been a fairly rudimentary understanding of the genetic and interaction influences with environment of childhood trauma, including the factors that regulate gene expression. And the most notable gene environment studies in PTSD and depression have implicated the FKBP5 gene, which is involved in HPA axis regulation. And so what those studies show and what they strongly suggest is that variants of this gene moderate the effect of childhood trauma on risk for disorders like PTSD and depression. So this was kind of the initial evolution in the field, but the landscape of genetics and epigenetics has since evolved. And we now know more about the pathogenetics of childhood trauma. And so this is what we know from systematic reviews and meta-analyses. We know that childhood trauma and maltreatment of itself is partially heritable. If you look at the SNP or genetic variant heritability for childhood trauma, it's around 6%. And there have been a few studies that have now used Mendelian randomization to try and establish causal inference between childhood trauma and mental health outcomes, showing that there is a potential causal relationship in terms of the genetic contribution between childhood maltreatment and adult mental health outcomes. On the epigenetic side, we know that epigenetic factors mediate the pathway between childhood trauma and common psychiatric disorders. And from global methylation and genome-wide methylation studies, a number of the same gene culprits have been identified, such as the serotonin transporter, such as the FKBP5 gene, which, as I mentioned, is one of the most replicated genes in the context of childhood trauma and adult PTSD and depression. And there may be a number of common pathways that may be the reason that this conglomerate of genes have a combined effect on disorders like PTSD and depression through pathways that are involved in the immune system and immune processes, neuroendocrine pathways, neural functioning, and so on. So to get on to the first of the three studies that I'm going to highlight, and the first is that of childhood trauma in adults with HIV. So this is the context in South Africa, but it is also context that exists in many other countries where HIV is highly prevalent. So South Africa is a country with a very high prevalence of HIV. About 9% of adults have HIV. Women are disproportionately affected by HIV, particularly in the 15 to 49-year age group. And HIV has a number of physical as well as mental health sequelae, both in children and adolescents, but also in adults. And up to a half of all people who have HIV and who are living with HIV experience neurocognitive impairments. And we refer to the cluster of neurocognitive impairments in HIV as HAND. So childhood trauma in conjunction with HIV can actually increase the risk of developing HAND. And some time back, now more than a decade ago, we were interested in testing this hypothesis that we refer to as the double hit hypothesis. And we hypothesized that HIV-infected women who are disproportionately affected by HIV, if they were exposed to childhood trauma, particularly moderate to severely affected by childhood trauma, they would be at much higher risk of neurocognitive impairments, but also at higher risk of the kind of direct sequelae of trauma, such as post-traumatic stress disorder and other common comorbidities like depression and anxiety disorders and substance use disorders. And what we showed was that there was indeed a double hit. And so the combined effects of childhood trauma in HIV was consistently associated with memory and executive function deficits, which one sees in HIV, but they were more pronounced in individuals who had early childhood trauma histories. And we also saw that there were structural brain alterations that were correlated with many of the neurocognitive deficits that we saw. And both the neurocognitive deficits and these kind of structural brain alterations were more pronounced in individuals with HIV who also had childhood trauma. Now, how might we explain this? I think that there may be common pathophysiological mechanisms, which I also highlighted at the outset, that there may be hypothalamic pituitary adrenal axis dysregulation in both HIV and childhood trauma, neuroinflammatory processes, as well as oxidative stress mechanisms. And we think these are the three main processes or three main common pathophysiological mechanisms that may explain this double hit. So we then went on to investigate some of the more biologically driven reasons for the changes that we were seeing. And this is the cohort that we have been following up for more than 5 years now. And so we have matched HIV-infected women with HIV-negative controls, and then also stratified them by the presence or absence of childhood trauma exposure. So as I said, childhood trauma is so pervasive in South Africa that it is very difficult to find a matched group of controls with little to no exposure to childhood trauma. And that just highlights what I've presented about the impact or the dual impact of HIV and childhood trauma on brain morphometry and neurocognitive impairment. So what were the genetic questions that we asked? So firstly, we were interested in whether childhood trauma and some of the genetic variants that have been implicated in cognitive disorders like Alzheimer's disease and other major neurocognitive disorders and lipoprotein E genetic variants have been sort of the variants that have been most extensively replicated and implicated as causal in Alzheimer's disease, for example. So that was the first question that we asked. Is there an association, and can we see this in relation to cognitive decline over time? So these are data from the 1-year follow-up, and what we showed was that this ApoE variant alone, as well as in combination with childhood trauma, was associated with poorer cognitive function. We also found interactive effects between this variant, the severity of childhood trauma, as well as a number of other variants in this viral protein R, which is an important viral protein in HIV. So we found an interactive effect with the decline in global cognitive scores over a period of 1 year. Then we also asked the question of whether there might be differences in telomere length. So telomeres are the caps on the ends of chromosomes and are associated with biological aging. And so you have an attrition of chromosomes with accelerated biological aging. And we wanted to identify whether telomere length shortening was a predictor of cognitive function in the context of HIV and childhood trauma. So we did not find an association here. The third question that we asked was whether there was an association between HPA axis genetic variants and neurocognitive decline over time in this population. And here again, we did show that there were variants in the corticotropin releasing hormone gene that independent of childhood trauma, but also one of these variants in interaction with childhood trauma conferred an additional vulnerability to neurocognitive impairments over time. So the second study that I'm going to highlight is one of post-traumatic stress disorder and its medical comorbidity, metabolic syndrome. And I go back to the slide that I showed where I mentioned that we need to, in addition to thinking about etiological factors, think about the mechanistic pathways. And so metabolic syndrome is a cluster of factors that contribute to cardiovascular disease risk. And metabolic syndrome, together with trauma exposure, childhood trauma exposure, cumulative trauma exposure, and post-traumatic stress disorder are non-communicable diseases that occur at high rates in our population. But if you look at data internationally, both post-traumatic stress disorder and metabolic syndrome have fairly high heritabilities and similar heritability. So the heritability for PTSD is around 40%. For metabolic syndrome, it's in excess of 40%. So they both have strong genetic components. And they both have relationships, which I'll show just now. They have bidirectional relationships. And there are also pathways that may explain the relationships between post-traumatic stress disorder and cardiometabolic disease that make it particularly pertinent to investigate genetic and epigenetic contributions. So in high-income countries, metabolic syndrome occurs in about a quarter of men and women. The prevalence in sub-Saharan Africa is in excess of 30%. And what has been found among South Africans is that those with metabolic syndrome actually have double the risk of cardiovascular disease compared to those without. And they also have a threefold increased risk of developing type 2 diabetes. And we have very high rates of type 2 diabetes in the country, especially among certain population groups. So among the Indian population, of which I'm a member of, and then the mixed race population, also referred to as the colored population in South Africa. Now, interestingly, childhood trauma increases both the number of metabolic syndrome symptoms, but also increases the risk of diagnosis of metabolic syndrome. So there is this complex relationship between childhood trauma, cumulative trauma, post-traumatic stress disorder, and metabolic syndrome. This is an indication of some of the putative pathways that have been that have been posited as explaining the links between PTSD and cardiometabolic syndrome. So in our study, which was referred to as Shared Roots, and these data that I'm going to present now were part of a doctoral study by a doctoral student known as Sylvanus Tokumo. And in the study, we were specifically interested in epigenetic modifications and DNA methylation, specifically in adults with PTSD and metabolic syndrome. So metabolic syndrome, as I indicated, is this cluster of factors, and the factors are those that are highlighted in the graphic on the right of your slide. And so you need this cluster of factors in order to make a diagnosis of metabolic syndrome. But the individual constituents of metabolic syndrome, the individual components, are also independent risk factors for metabolic syndrome. So the question that we asked here was whether there was an association between childhood trauma, post-traumatic stress disorder, and metabolic syndrome, and whether we could see these changes in terms of DNA methylation. So DNA methylation is, as I said, too, an environmentally sensitive epigenetic mechanism that can influence gene expression, and it can produce long-term effects in individuals with post-traumatic stress disorder and metabolic syndrome. So prior to conducting this study, there had been some DNA methylation studies in PTSD and in metabolic syndrome independently, but not in the comorbidity. And these were some of the genes that were identified as being either hypermethylated or hypomethylated. So hypermethylated in red and hypomethylated in yellow. But as I said, there had been no studies investigating the epigenetics of PTSD in conjunction with metabolic syndrome. So we conducted this genome-wide study. And we asked the question of whether there was an association at an epigenetic level between PTSD and metabolic syndrome. And we also stratified our sample by exposure to childhood trauma. So what you see here is a relatively small sample study, but it was essentially a proof of principle study. The groups that we conducted the investigation here are shown in terms of their demographic characteristics, and these were demographic and cell type data that we had to adjust for in our analyses. So there were no significant differences in age, in sex, and in cell type composition across the groups. And so in this epigenome-wide association study, and I should have said this when I explained the focus of epigenetic studies, but one actually investigates differences at CPG sites. And so this is what we examined. And what you see here are some of the hits that reached epigenome-wide significance. So in the red, it's those genes that were hypermethylated, and at the bottom, those genes that were hypomethylated. So our study then showed that there were genes that were commonly methylated, either hypo or hypermethylated, in this instance, genes that are involved in the immune system and energy metabolism for PTSD and metabolic syndrome comorbidity. So as I mentioned, this is a small sample. It was a proof of concept study. It needs replication, and the findings also require validation in other populations. But I think it provides initial evidence that there are shared epigenetic signatures in PTSD and metabolic syndrome. So the last study that I'm going to highlight is in the context of rape trauma, and this is the Rape Impact Cohort Evaluation study, which is still ongoing. And this was also a doctoral student project conducted by a student, Yanni Notlam. Now, I said at the outset that rape is the most pathogenic trauma for post-traumatic stress disorder. And in fact, studies have shown that the incidence at 3 months post-rape of PTSD is up to 35 to 45%. And women who are at higher risk of rape trauma are also at much increased risk of developing PTSD compared to men after rape trauma. But that is true for other types of interpersonal trauma. So across the board, women have at least a twofold higher risk of developing PTSD compared to men. Now, prior studies have had limitations in terms of the design of the studies. So few have comprehensively investigated risk and protective factors for PTSD longitudinally, especially in the post 3 month period. So I said that, the incidence is 35 to 45% when one assesses for PTSD, but few studies have actually examined the trajectory of PTSD post 3 months. And there's also a lack of epigenome-wide association studies investigating differential DNA methylation in relation to PTSD in African populations, and no studies prior to this one in rape-exposed women or in rape-exposed populations generally. So this was a study in rape-exposed women, and these were women who were recruited very early after rape. So they had reported a documented rape incidence in the past 20 days prior to being excluded in the study. And we recruited rape-exposed women as well as controls. And what you see here are the numbers that were retained over time. And this shows data up to 36 months, but we are now further along in the study. And we have been gathering data on mental health, but also physical health outcomes. The main research question that was asked in the study was about the incidence of HIV in the context of rape, because you may find this surprising, but even though South Africa, is referred to as the HIV capital in the world and also referred to as the rape capital in the world, we actually had no data, no population-based data on the incidence of HIV following rape. So I think this is an indictment, and it says much about the focus of research on rape and on HIV, and that these lines of research have actually occurred in silos rather than alongside each other. So we collected much kind of physical health data, biological data, mental health data, and biological samples, including genetic samples. So what I'm going to present here are just 3 of the aims that formed part of this doctoral dissertation. The first was to identify some of the risk and protective factors that affected the symptom trajectory of PTSD post-rape. And we saw this as a necessary first step, because if you are trying to identify, genome-wide changes in PTSD, it's important to do this alongside having a good understanding of some of the environmental and other psychosocial factors that affect the symptom trajectory of PTSD following rape. So that's what we wanted to do as a first step, and then identify these genome-wide differentially methylated genes associated with PTSD, and to investigate this longitudinally using an EWAS study, so an epigenome-wide association study approach. And then we wanted to investigate whether changes in this gene that has been so replicated in the context of childhood trauma, but also in PTSD and depression, was related to PTSD symptom change post-rape. So this is a description of the design of the studies that I'm going to very quickly cherry pick findings from and highlight. To say that we found that the incidence of HIV in women who were raped was in line with our expectations. So as you can see that at 3 months, there was no real discrimination between the rape-exposed women in the purple line and the non-rape exposed women, but that over time, you can see the separation in terms of HIV incidence. And so this is, I think, the first indication in our setting of the importance of screening women for HIV in the long aftermath of rape. So we have these rape centers around the country, which are kind of one-stop centers for women who report their rape, where they will receive both medical, forensic, as well as mental health support, although the mental health support is minimal. But many of those women are lost to follow-up. So, they are not followed up over time, and there's no retention over time. And I think this highlights the importance of ensuring that we don't lose rape-exposed women to follow-up. Now, if we look at the post-traumatic stress symptom trajectory following rape on the left, you will see the difference in the PTSD symptom profile. And so this is not PTSD diagnosis in a woman who reported rape and women who did not. And you can see that, over time, PTSD symptoms still remain high. So this is the Davidson Trauma Scale. 40 is the 39 to 40 is the cutoff. And you can see that, 24 months, many of these women will actually meet the cutoff on the DTS, and I'm referring to rape-exposed women for PTSD. And then I show out of interest the proportion of women with suicidal ideation and the trajectory in rape-exposed women in purple versus the non-rape-exposed women, also showing that, differences in relation to suicidal ideation. And this is not suicidal ideation with intent only. It is any suicidal ideation that is endorsed over time. So what we showed in terms of trying to identify some of the risk and predictor factors was that rape stigma was a very important predictor of PTSD scores. So these were data over 6 months only, not over the 36 months of the study. And so women who have high levels of rape stigma, and rape is a very stigmatized trauma, in our setting, but it is a stigmatized trauma all over the world. And so I think it's important to assess for rape stigma and to manage it in the early aftermath of trauma. We also showed that baseline depression scores actually predicted PTSD scores at both 3 and 6 months post-rape. But that baseline resilience, social support, perceived stress, and alcohol use were actually not significant predictors of PTSD scores over time. So these are data over 6 months, but we are now analyzing data over a longer period of time. So these are just a graphic representation of depressive and PTSD symptoms, and then the interaction with rape stigma over time. So the second aim I am going to very quickly highlight, because I see that I am reaching the end of my time allotment. And as I said, this was an investigation of genome-wide differentially methylated genes that were associated with PTSD, but also the longitudinal change in methylation of these genes in women who have been raped. So without going into too much detail, I want to perhaps just highlight that in our study, we did identify genes that have been identified in genome-wide studies in PTSD. For example, this gene that I have mentioned that may, to some extent, explain the high rates of PTSD in women, or the greater vulnerability of PTSD in women compared to men. And we also showed a change in methylation of genes over time in rape-exposed women. And we showed that there was an association between the kind of trajectory, the direction of PTSD symptom change over time, as well as methylation in some of the CPG sites in this particular gene, which, as I said, is a pituitary adenylate cyclase gene important in HPA axis function. So to sum up then, the epigenetic findings point to two key genes and differential methylation in these genes. In addition to this gene that is associated with a dysregulation of HPA axis function, this second gene that we identified that was differentially methylated is associated with adverse neuronal development and dysregulated blood glucose levels. And while I don't present findings here, another doctoral student is focusing her investigation on PTSD trajectories in relation to metabolic syndrome in this sample. And that is not an epigenetic study, but what we plan to do going forward is to see if we can replicate these findings by specifically investigating the comorbidity of PTSD and metabolic syndrome in this sample. So then our last aim in this set of investigations was to specifically assess for methylation in this particular gene and at a particular site in this gene, FKBP5, also very important HPA axis function, and to assess whether changes in methylation were also correlated with PTSD symptom change over time. And so we did find that there were associations between methylation, so hypermethylation and particular symptom profiles over time, and then increased methylation and specific symptom profiles over time. So this is also, I think, proof of concept study indicating that in women who have been raped, there are differential methylation of genes that have been found in GWAS studies, but also in other epigenome-wide association studies in individuals with PTSD who have other types of trauma. And I think they provide evidence that these epigenetic changes can persist over time and can track the course of PTSD symptoms over time. not just PTSD symptoms, but also depression and potentially other comorbidities. And this is why we are interested in investigating PTSD and metabolic syndrome in relation to these methylation changes that we have shown here. So in summary then, childhood trauma is a distal exposure for other cumulative trauma. And if we think of childhood trauma as being the first hit that sets individuals up for consequent trauma exposure and then for adverse sequelae, I think it underscores the importance of always thinking about childhood trauma in any study that one is conducting in the realm of genetics and epigenetics. So childhood trauma and PTSD have unique epigenetic and genetic motifs, but they also have shared motifs. And we're still at an early stage in trying to distill pathogenetic mechanisms of PTSD and its many common comorbidities, including physical comorbidities such as HIV and metabolic syndrome. But I think that it is really important to try and identify these genetic and epigenetic signatures of comorbidity, because typically we do these case control studies and we screen out individuals who have comorbidities. And that doesn't reflect what we see in real world practice. I mean, for those of us who see patients with PTSD or patients exposed to trauma, we know that PTSD is just one sequelae, and many patients only present for treatment, and that is certainly true in our setting where patients present late. They usually present late when they have many other comorbidities. But I think that the field of genetics, epigenetics, omics is moving fast along in PTSD. It's a very exciting time. And I don't think it will be too far along before these findings will be able to inform. I mean, they won't be able to inform alone, but I think that they will form part of our armamentarium when we think about more patient-tailored treatment approaches. So thank you very much for your attention.
Yes, thanks very much, Soraya, for this content-rich and very interesting